like to introduce our first speaker of the day, Dr. Nick Johnson. Dr. Johnson is the Associate Professor of Neurology and Human and Molecular Genetics and Vice Chair of Research in Neurology at Virginia Commonwealth University with a focus in inherited neuromuscular disorders. He received his undergraduate degree in molecular and cellular biology and psychology at the University of Arizona. He then obtained his medical degree at the University of Arizona. He completed his neurology residency and combined fellowship in neuromuscular medicine and experimental therapeutics at the University of Rochester. He leads the GRASP LGMD consortium, which focuses on developing clinical outcomes and biomarkers in the limb-girdle muscular dystrophies, and co-leads the Myotonic Dystrophy Clinical Research Network, which focuses on developing clinical outcomes and biomarkers in the myotonic dystrophies. His laboratory is focused on identifying the pathogenesis and progression of myotonic dystrophy and biomarkers in limb-girdle muscular dystrophy. Dr. Johnson conducts therapeutic trials in many other inherited nerve and muscle disorders, including limb-girdle muscular dystrophy and facioscapular humeral muscular dystrophy. Thank you, Dr. Johnson, for being here today, and I will let you take it away.
Thanks, Michelle, for having, I froze there first. Okay, thanks, Michelle, for having me. I'm excited to be here and kicking off what looks like a great day of speakers. And I think, yep, you should be able to see my screen. So I'm going to kick us off by talking about research in clinical trials in limb-girdle muscular dystrophy, which is an important topic and really I would consider it to be a hot topic at this point, as I'll explain in a little bit. So there we go. These are my disclosures. So I will be, we have received research funding from Sarepta Therapeutics, from ML Biosolutions, and from Edgewise Therapeutics, and I'll be speaking about some of those projects today. But I thought it would be remiss if I didn't start with at least a short 10,000-foot overview on limb-girdle muscular dystrophy as a whole, because that's the foundation. Hopefully, you're here in the right day. So limb-girdle muscular dystrophy, the estimated combined prevalence is about 1 in 20,000. So as a group, it makes it the 4th most common cause of muscular dystrophy. It has a really complicated naming system that has gone through a revision that I'm not sure it helped to make it any less common. But to break it down, if you have an autosomal dominant, which means that every generation has the disorder and you only one bad copy of the gene to cause the disorder, those are called type D, or they used to be called type 1. If you have a recessive version, which means that you need one bad copy from each parent and 2 bad copies causes the disease, that's type R. It used to be called type 2. With a new nomenclature, which is the type D, if you know the genetic mutation, then you can put that little number behind it, and that tells us what gene mutation is. So LGMD D1, which is caused by a single mutation in the gene called DNA JAB6, the most common dominant type. Type 2 autosomal recessive, there's 27 different forms at this point. Calpain 3 or LGMD R1, the most common, followed very closely by LGMD R2 or 2B caused by dysferlin. So really complicated naming system. I wish it was any simpler. It causes all of our medical students to stumble over it. But when you hear your doctor ask you, do you have R1, R9, that's what they're asking about. As a combined group of disorders, these are slowly progressive. They cause symmetric weakness, and that weakness is in those red muscles that you see highlighted here. So the shoulders and then the hips, although it can progress to other muscles, of course. It's beyond that, it's variable. So some forms of limb-girdle muscular dystrophy affect the heart, some do not. Some affect the breathing muscles, some do not. And in some cases, the weakness can start more in the hands and feet versus in the hips and shoulders. And that's really gene specific. It is exceedingly uncommon that limb-girdle muscular dystrophy would affect the extraocular movements, so the muscles that move your eyes, or cause facial weakness. And typically does not cause any type of intellectual disability as an entity. But those are some kind of hallmarks. The picture that you see there is the outside of your skeletal muscle membrane. And so that's composed by a bunch of scaffolding proteins. And essentially, if you mutate or have a problem with any one of those proteins, that can cause a form of muscular dystrophy, most commonly limb-girdle muscular dystrophy. And we're going to come back to that when we start to talk about therapies. But that's really the primary cause of limb-girdle muscular dystrophy. We live in an era where genetic testing for the limb-girdle muscular dystrophies is very simple. In fact, it is free. So there's, from that standpoint, it's really accessible for people to get genetic testing. And this is a result of about 4,000 or so individuals that were part of a large next generation genetic testing sequencing program. And as I said, you can see calpain-3 or LGMD1, the most common, very closely by dysferlin, and then the rest kind of in descending order from there. I'll come back to this later in the talk, but one of the new challenges of the limb-girdle muscular dystrophies in terms of diagnosis is that as easy as it is to get genetic testing, it's just as hard to figure out how to interpret it. So of the people in that pilot study, 4,000 or so individuals, only about 24% of them were truly negative, and about 72% had a variant of unknown significance. I'm going to talk about that when I talk about the work that's going on in our consortium. So that's a new challenge. As I said, the genetic basis of limb-girdle muscular dystrophy, at least 28 different gene mutations as causative. It is not X-linked, so it's common in both men and women. We talked about how it's inherited, dominantly or recessive, and you can see a table breakdown of that of those different gene naming systems. And at the very end of the day, I think you're going to hear from Paul at the MDA, who, as a result of this, is able to give us better clarity on terms of our diagnostic codes, coding in the United States here. So how do you diagnose limb-girdle muscular dystrophy? Well, LGMD is progressive, and the severity of symptoms and age of onset may vary among the subtypes. Some cases are mild and more severe. It doesn't matter what particular mutation it is. That's true across the board for any subtype of LGMD. The CK, which is a level of your muscle breakdown, may be elevated. Sometimes people in the past would have done a muscle biopsy to show degeneration, regeneration. But really the first line now is to do that large next generation sequencing panel. Okay, so that's LGMD in a nutshell. I know we zipped right through that, but hopefully most of you on the phone already had some knowledge there. And I'm going to move to talking about clinical trials and getting ready for this next era of LGMD research that we're just stepping our toes into at this current point in time. And when people talk about preparing for clinical trials or getting ready for clinical trials, oftentimes they'll show a straight road going down, but in fact, this is really what it looks like more. You have multiple different people and areas, and they all have to come together at the same place to allow for a clinical trial to be successful. So hopefully, with your support, that we're getting to a place where we're ready to merge all these lanes into a single path and get down the road on a clinical trial. So if you have not participated in research before, I would strongly encourage you to do that, or think about it at least. You can look for studies related to limb-girdle muscular dystrophy on clinicaltrials.gov, So website's right there. I'm going to talk about some studies as we go, but if you have any curiosity or want to know about other things, you can always go to that website anytime. It's updated pretty regularly. Oftentimes that if you type in LGMD, you'll get a list. It'll say whether or not the study is seeking new people, or if it's closed, or it's not started yet. And it usually includes some information about how old you have to be, what type of subtype of limb-girdle you have to be, and whether or not you need to be walking or not walking. And most importantly, it will always include contact information. So if you're looking at it, you're not sure, and you're not sure about the risks, and you don't want to know, that's what that contact is for. Just either email the person or call them. That information is there for you. Say, hey, do I qualify? Do not qualify? Most of the time, you'll get an answer back pretty quickly, and you'll know the details. So, as I said, why do we need to prepare for clinical trials now? Well, first of all, our ability to diagnose LGMD has improved, and the cost of commercial testing has lowered to either $250 if you pay for it, or at this point, it's now actually, it's really free. But also the number of individuals with variants of unknown significance or negative testing is unknown. So those people over there, over 50% with unresolved variants of unknown significance, you don't have a diagnosis of limb-girdle muscular dystrophy, and you don't not have a diagnosis of limb-girdle dystrophy. It's kind of in limbo, which is a real problem when we think about these gene therapies that we'll talk about. And speaking of which, at the same time, advances in gene therapy have identified LGMDs that are top candidates for gene replacement therapy. I'm going to end my talk with this. And so across the board, researchers like myself, our consortium, and companies are developing other molecularly targeted therapies. And I think there's a real possibility of at least not one, but several clinical trials on LGMD happening over the next 5 years, hopefully sooner. But the pace of therapeutic development has outpaced our efforts to prepare for clinical trials. So as quickly as people are coming up with new therapies, it's great. It's awesome, but we're not ready to do those trials because we don't know, all the information we need to be successful there. And these are the gaps that we need. So we need the tools to measure response to therapy in a clinical trial. So we need to know, what percent change on a muscle strength test is clinically meaningful to you, and is able to be measured precisely. How what's your experience living with limb-girdle muscular dystrophy? And then finally, how do we diagnose people with LGMD? And this is a big problem. And there's, as I said, there's over 28 different types of limb-girdle muscular dystrophy. If one person tried to solve this at one subtype, it would be decades until we got all the way through all the limb-girdle muscular dystrophies. Not enough. Needs to be faster, right? Because we just said that we've got these cool new therapies coming. So we need people to come together, think outside the box, ways to measure LGMD and new avenues for treatment. And we need those people to work all together. And in fact, we know from other diseases like the Duchenne muscular dystrophy or spinal muscular atrophy, if those people all sit within the same tent, it will move faster. And so one solution is to create an LGMD clinical research network, which is what we did. This is the GRASP LGMD Consortium. This is an international group of academic medical centers seeking to develop outcome measures and biomarkers for clinical trials, understand the disease impact, improve the diagnostic journey, and to train the next generation of researchers. And the goal, of course, across the board is to speed therapeutic development in limb-girdle muscular dystrophy. These are the consortium sites. Hopefully you live close to one of them or within driving distance. We're always adding new sites. So if you don't, you can check in. My contact information will follow here and say where you live, and we'll see if if we're adding sites in that space. So this is coordinated out of VCU, which is my site. Jeff Statlin at KUMC is our co-coordinating center. Chris Weil at Washington University is our supports our biorepository. And then the physiotherapists at Nationwide Children's Hospital are our lead evaluators for the clinical outcome measures, which we'll talk about, but the sites you can see there across the United States and Europe. And these are the overview of the current studies. These are all natural history studies, so they're designed to capture the individual experience living with limb-girdle muscular dystrophy and to test how best to measure that over a period of time. You can see that individuals with calpain-3, ANO5, dysferlin, or DNAJB6 can enroll in the defining endpoint study, which is a 12-month study. There is individuals with sarcoglycan. Any type of sarcoglycan can enroll in the second arm of that same study, 12 months. We have completed enrollment for our LGMD2I or R9 FKRP study, and I hope to have results to share with this group in the future. And then finally, there's a new Becker's study that's enrolling over the course of a 12-month period. So if you have one of these mutations, please do contact us. I'm going to give you that information and consider participating in these studies. Because these studies help us close the gaps to treatment trials. So they're going to take all that mess of roads, all the different information, we're going to try and test it with the express goal of designing good treatment trials that give us the information that we need. So that information in terms of a treatment trial, I hope is that that drug works, but we also need to have the information to know whether or not the drug is not going to work. And one of the worst case scenarios for our community is that if the answer is maybe yes, maybe no, because then that drug just kind of sits in limbo for a long period of time and never quite gets fully approved, which is a real challenge for the community. So let's talk about those gaps. So these are the tools for clinical trials. Outcome measures is a term that you'll hear frequently. They are the tools that we use to tell if somebody with limb-girdle muscular dystrophy has changed between 2 points in time. And if there's something that's measured in the blood or the muscle, they're called biomarkers. If there's something measured by clinical evaluators or physical therapists, they're often called clinical outcome assessments. So that's strength or ability to do tests. And they could also be questionnaires that you fill out to talk about your experience of limb-girdle muscular dystrophy. And when we were designing these studies and thinking about outcome measures, these are the elements that are important to us. So most important to everybody is they have to be pertinent to limb-girdle muscular dystrophy. It's all well and good if we do a test that asks how well you're thinking. That's not the problem that's caused by limb-girdle muscular dystrophy. We want to focus on the things that are caused by the condition. They need to be reliable, so the more stable they are over time. Each time you measure it, the fewer the number of times you need to measure it, frankly. They need to be sensitive to progression in limb-girdle muscular dystrophy. One of the challenges we have is that it is a progressive condition, but it is slowly progressive. And I think we're looking for things that we can measure over 6 months or a 12-month period and detect that change. Because same point as always, we want those trials to be as short as possible so that we can get to the answer about whether or not that therapy is going to be effective. They should be inexpensive and they should be feasible to collect at multiple sites. So you can see the number of sites, everybody has to be able to do it. So it can't require too specialized of tools or instruments. So you need the right tool for the right job. So these are, measuring gene expression, for instance, doesn't tell you whether or not you're experiencing pains. And different tools are very important at different times in the drug development process. And so we're looking for not just one tool, we're looking for a clinical trial toolbox that we can give to our colleagues in the pharmaceutical industry and say, for this trial, this is what you might want to use. For this trial, this is what you might want to use, depending on the therapy and the condition and the point at which that drug is in development. But in limb-girdle muscular dystrophy, one of the biggest ones is strength and function. So we have many standard techniques for measuring muscle strength. So manual muscle testing, this is what your doctor already does when you go to a clinic. We can make it more quantitative, which is that quantitative biometry you see in the second panel. And then we can look at dynamic strength as well. But many standard measures for things like how far you walk, get up from the chair, go up from stairs, and how far you reach. And we can also combine these tasks into composites, but there's still an awful lot of work that needs to be done. So we need to standardize our outcome equipment and procedures, train our evaluators, and understand the relationship between the age, the men and women, relationship to different gene mutations, and then how somebody comes as they start the study. But here are some examples that come from the LGMD 2A or R1 cohorts that suggests that one outcome measure that we're particularly keen on, the North Star assessment for limb-girdle muscular dystrophy, is both reliable to use and does detect difference between people who are walking and not walking. So you can see the testing from day 1 to day 2 for this particular outcome measure is highly reliable. It's the same each time you test it. And that the difference in the score between those people who can walk here and those people who can't walk is different. And so that helps us say that this outcome measure is measuring something like ambulation or the ability to walk. And we think that that type of thing is really important in limb-girdle muscular dystrophy. Hope you do too. Okay, so for biomarkers. For gene therapy, the most obvious biomarker is expression of the missing protein, which is what they use in Duchenne muscular dystrophy. So you can see this panel at the bottom there. So you've got this this control muscle here, which just should look like a kind of a black square for you. There's secretly muscle fibers behind that, and you can see that in this particular trial, after you've replaced the beta-sarcoglycan gene using AAV technology, you can see that that expression of that protein has come back to the muscle. So that's pretty good. And that's probably important. And I think most people would agree that that says that that drug's probably working. And so that's probably one of the most obvious kinds of biomarkers. But for some limb-girdles, we talk about measures of evidence of muscle destruction like CK or inflammatory markers. And then one of the newer outcome measures that's being tried is MRI, which is a lot less invasive than a muscle biopsy. Although I will tell you that when we speak with our focus groups, people have mixed opinions about whether or not they would have a muscle biopsy, MRI, as part of a study. So I'm going to leave that for you to think about. And so this is an example of a leg with limb-girdle muscular dystrophy. And you can see the bone here, and this is a normal muscle here. And these muscles over here, you can see the percentage there. They show some percentage of fat replacement of that muscle. And this muscle here, 64% fat replacement, pretty different looking than that healthy muscle. So that's definitely another way that you can measure change in disease over time. And it's probably more sensitive than, for example, how fast you walk over 30 feet. Lots of work to do. So we got to make sure those are reliable, responsive. It's the same thing, biomarker, clinical outcome assessment, same work that needs to be done. And the second gap here is the patient perspective. So we take it really seriously that what we're measuring in our studies and what clinical trials we'll use in the future matches your perception of the disease that you experience. So we are in the process of developing a disease-specific health inventory, which is a questionnaire that would be used for clinical trials. Some of you on the phone have probably taken some version of that survey from us, so thank you. And we started with patient interviews. So we said, well, what matters to you? Pretty open questions. And then we did a larger survey to see whether or not those different items were validated. And so these are the results of those patient interviews. So you can see, not surprising, if you count the number of quotes for different themes, so these different ideas here, problems with mobility and ambulation, number 1 issue. Inability to do activities, number 2 issue. So that's not really very surprising to us. You do notice some change in how you function in society and some stress as it relates to those problems, which again is not surprising. And we hope that as these treatment trials get underway, that we're able to improve some of this stuff across the board. Here's a result from that survey, which, a 4 is the most impact that a particular item may measure. And this is divided cross-sectionally by age. And so you can see, again, problems with hip, thigh, or knee weakness or difficulty with ambulation are very frequent, and then they do progress as age gets on. But again, problems with shoulders and arms and inability to do activities also very important, impactful. In terms of the population impact of the social and cognitive health, again, you can see that fatigue is very important, and then as is a change in your body image due to disease. So hopefully these continue to be used in clinical trials, and it's our way of capturing an individual's perspective on the disease and whether or not that treatment is impacting the things that they think are important. Okay, so gap number 3 is improving our genetic diagnostics or our testing for limb-girdle muscular dystrophy. So because of the possibility for gene replacement therapies and to help guide medical surveillance, we feel very strongly that anyone with progressive weakness in your hip or shoulder girdle needs genetic testing. That's an important step because many of these therapies that we'll talk about in the back half of this lecture are gene-based therapies. And so without a positive genetic test, that individual will not qualify for those studies. We mentioned this before. So good news is about a quarter of people or so end up with a confirmed diagnosis. About a quarter or so are completely negative. So that means this is the wrong set of genetic testing. Something else needs to be done. But that 55% is really bothersome to us because those are people who are left hanging as a result of this, and we need to do better. So for a variant of unknown significance, what's next? So we are very good at identifying them, but not particularly good at resolving them as it stands right now. So here are some ways that we think about whether or not you can resolve that variant of unknown significance. So does it track with affected individuals in your family? So for example, if you've got a brother or sister also with limb-girdle weakness, do you both have the same variants of unknown significance? That's helpful in terms of determining whether it's disease causing or not. We can look in the tissue. So this is another way of looking at that. Now this is alpha-sarcoglycan, and you can see that in this particular individual, there's no alpha-sarcoglycan expressed in the muscle. This is a control showing you that nice ring around the muscles, what you would expect to see. Or you can use functional assays. So this is DNA JB6. This work's done in Chris Weil's lab at Washington University, and you can see that whether or not that particular mutation is damaging. And then sometimes we can look at the RNA to see whether or not expression fits. But this is time consuming and it's done very differently across the country. And so it needs to be adapted to each person's situation. And so folks within our consortium, Chris Weil is leading this effort as an ability to improve these genetic testing panels. And so this is a partnership between geneticists, clinicians, researchers, and patients. It's NIH supported. And there's this group is working on gene-specific guidelines for limb-girdle muscular dystrophy, focusing specifically on calpain-3, dysferlin, A105, FKRP, and the sarcoglycans, because those are the ones where therapy is most imminent. You can do this. You can help. So you can volunteer with the ClinGen LGMD expert panel. You don't need to know anything. So you could do this if you're a patient, a caregiver, advocate, and the email person in contact is Chris Weil. His email is down there at the bottom. We'll see it. Wash you. You can see it there. And that's one good way that you can think about helping to improve genetic testing. You can do something. All right, so clinical trial preparedness is a collaboration, industry, academics, basic scientists, advocacy, and people with LGMD. Our GRASP LGMD consortium focuses on those efforts to help us get across the board, understand the natural history, and really prepare for these upcoming clinical trials, and create a group of sites, evaluators, and clinicians who are focused on carrying those efforts out. So you can participate. So you can contact me. There's my email. You can contact our network coordinator, Mary St. Romain. You can see her email. You can find our studies at clinicaltrials.gov. You can look on our website, grasplgmd.org. Anything that you, those are all different ways to all get back to the same two people, which is Mary St. Romain and myself. Okay, so what's all that effort about? Why am I pushing so hard on getting those clinical outcome assessments up and running? Well, it's because there are really exciting therapies in development, and I think I'm incredibly optimistic about where LGMD is going over the next 5 to 10 years. So probably the biggest deal here is gene replacement therapy. What is that? So that is where you use a virus, you take the part of the gene that's missing, you stick it in that virus, and then you infuse that virus into the person to allow the missing piece of DNA to get back into the cells that need it. So these are best done in those autosomal recessive conditions because you're missing something, and the gene replacement therapy helps replace that. Because they use viruses, these are likely one-time therapies. So I know there's a lot of research that will allow people to get redosed in the future, but as it stands right now, these are one-time therapies. And the biggest risk of any one of these gene replacement therapies is the immune reaction that might occur as a result of the infusion. That's where all the effort and safety reporting is taken in that first 30 to 60 days of the gene replacement therapy. I showed you this at the beginning. And so many limb-girdle muscular dystrophies are caused by problems with that the dystrophin-associated protein complex or membrane repair proteins. These are the different limb-girdles that are involved in that particular scaffolding. And every single one of these is amenable to gene replacement therapy, shown on this screen. The way to think about it is that that protein complex is like the scaffolding on a skyscraper, and so when the skyscraper goes in the wind, it can sway, it doesn't break, but because when one of these proteins are missing, that's part of that scaffolding is weak, and it leads to collapse of the muscle wall. Here's some examples just to show you. So this is work that was done by folks that are now at Sarepta Therapeutics. And this is looking at a beta-sarcoglycan gene therapy. So you can see, again, in these mice that are missing that protein, that there's no expression of it on the muscle wall, in that when you return expression of that protein through this AAV gene replacement therapy in a mouse, you can see restoration of the protein wall. So that's good. Wouldn't do anything if it didn't help you feel better. So this again is that same mouse experiment now showing you that the knockout mouse is weak, and that when you replace the gene therapy, it gets stronger and also has less fatigue. It's not just for beta-sarcoglycan. This is work in LGMD R9 or 2I, FKRP, and here, same idea. You can see that reduced expression in mouse models that have some version of a FKRP mutation, and that when you restore that using AAV9 FKRP gene replacement therapy, you can see that the muscle protein is restored. And again, same thing, wouldn't do anything if it didn't improve your strength. And here's looking at running distance, which is another way showing you that the running distance is improved with that gene replacement therapy. So pretty exciting across the board. These are the current gene replacement therapy programs that are in preclinical or early clinical development. So that's beta-sarcoglycan, alpha-sarcoglycan, gamma-sarcoglycan, ANO5, dysferlin, calpain-3, sorry, I missed the 3 there, and FKRP. So every single one of these is associated with at least one, if not more, companies who have indicated that they intend to pursue clinical trials using an AAV gene replacement therapy in those particular conditions. And in some instances, for beta and alpha sarcoglycan, they've had at least early phase clinical trials where 3 to 6 people have received a gene replacement therapy at a single center. Okay, it's not the only way. I think most of us would say we don't want to put all our eggs in one basket. So there, outside of gene replacement therapies, there are small molecule approaches. And small molecules are like any other drug that you've ever taken, Tylenol, aspirin, that blood pressure, cholesterol medicine you might be prescribed by your doctor. The advantages of these is that they're usually pretty easy to give, so they're often taken orally. They often act on downstream actions from the loss of a protein. So there's probably more traditional trial and approval pathway from those gene replacement therapies, but they're have equal possibility of benefit. And the one that's furthest along is for 2I or R9, which is ribitol, which here you can see in FKRP. The problem is that you have these series of enzymes. They're supposed to add sugars to the end of that skeletal muscle alpha-dystroglycan protein complex. And here, what they're trying to do is just give more of that ribitol to allow it to get over the hump that's caused by that poorly acting FKRP enzyme in LGMD2I or R9. And that study is now in phase 2 clinical trials. And so I would expect to see that continue to move through the pipeline. We've spoken quite a lot about gene replacement therapies or the recessive forms of limb-girdle muscular dystrophy. And that's because the gene replacement therapies are furthest along, and that small molecule that I just spoke to is already in clinical trials. Stay tuned if you have dominant form of limb-girdle muscular dystrophy. Stay tuned to this idea, which is antisense oligonucleotides. So these are artificial pieces of RNA that can knock down gene expression. And in the past, they've been used in spinal muscular atrophy, in Duchenne muscular dystrophy. There's even now early clinical trials in myotonic dystrophy thinking about these particular compounds. And they haven't been tried yet in limb-girdle muscular dystrophy, but for things like LGMD, D1, or GNAGB6 mutations, there is a possibility that this type of approach may be effective. Unfortunately, I think that's still a few years down the road, but it's something to keep an eye on in other neuromuscular conditions. And I would be remiss if I'm talking about therapies to talk about gene editing technologies. This is the Star Trek version of the gene replacement therapy that I spoke to. So the gene replacement therapy, that virus particle, the DNA that's provided, never gets into your genome or your master code. It just sits in the cell. Same thing like if you are chicken pox, you've still got bits of that varicella DNA that are just kind of sitting there in the cell and not being part of the actual genome. The nice thing about that is that they can get kicked out of the cells. They're not going to be passed on to your kids, for example. But gene editing is where we use a tool called CRISPR or Cas9 therapy, or excuse me, a CRISPR Cas9 enzyme. And you actually, it's like a pair of scissors, and you can remove the defective gene and put the piece in that's effective. So if it's defective, you're putting something in that's effective. And so the biggest issue there is that it's all a safety issue. So this technology is readily available. We use it in our lab. We use it in mice or in cells. So it's not that the technology is not available, but the biggest concern is to make sure that that gene clipping doesn't occur in other genes that are equally important. And so that kind of safety stuff is still, it's ongoing. I think this is something that's maybe 5 to 7 years away, but this is probably, at the end of the day, this is going to be, if the safety signal is able to be tolerable, this is the kind of thing that's going to be really quite effective in terms of treating the condition. So in conclusion, there's been significant progress in drug development in the last 5 years. I'm as anxious as you are to get these trials up and going. We do need to have patients as early phase trials begin. One of the biggest letdowns in the community is that, there's lots of people out there with LGMD2I or R9 that would be capable of participating in the clinical trials, but those early phase clinical trials often only take, just a handful of individuals at select sites. And so that's important because that's selected to test whether or not that drug is safe. And then hopefully as a phase 3 trial begins, it becomes more widely available to others in the community. So we just need a little bit of patience because it feels like we're right on the cusp, but just not quite out of the starting gate yet on so many of these different subtypes. We're working as hard as we can to develop tools to conduct clinical trials. We need your help for that. We can't do any of this work without individuals with limb-girdle muscular dystrophy working with members of our consortium and the companies that work with our consortium to make sure these trials are designed for you, to buy you for you, and that they're able to show a benefit as soon as possible, because that's what's going to get that drug approved and accessible to everybody with that subtype of limb-girdle muscular dystrophy. Couldn't do it without our supports to the MDA, National Institutes of Health, Coalition Cure, Calpain-3, ML Biosolutions, Sarepta, and Edgewise Therapeutics. And a special thanks to all of our investigators who are working hard on this and across so many different things. I have bold highlighted there Jeff Statlin at KUMC, who's really a key collaborator and helps me greatly in terms of keeping this consortium up running and making sure that we're getting you the tools and answers that you need. And with that, I will pause and take questions.
Thank you, Dr. Johnson, for that great presentation. I would now like to open the symposium to any questions the audience may have about research and clinical trials. Like I previously mentioned at the beginning of the webinar, if you hover over the bottom of your screen, you will see a Q&A bubble. Click on the bubble and type in your question. And we will just wait for them to come in. In the meantime, I wanted to ask you, Dr. Johnson, a little bit about barriers to clinical trials. So maybe English is maybe not be their first language, or maybe they live in a rural area. How can we make sure that those individuals who are interested in clinical trials get to be a part of one?
Yeah, that's a great question. So, so the language is an easy one because we can and do translate our studies into Spanish for sure, and really any other language. There's always an interpreter that's available. So, hopefully, we've solved that barrier for individuals. The geography issue is a big deal, and I respect that as being challenging. I think the best that we can do is to always add trial sites at places where they're going to be accessible to other individuals. We're a little short on the West Coast of the United States, I acknowledge that. We did try during the midst of the COVID-19 pandemic to try and do some of these outcome measures at home. What we found actually is that unfortunately, we were still much more successful at doing them in the clinic. And so we try and make it as easy and accessible as possible to people.
A few questions have come in from our attendees. How are candidates selected for clinical trials?
If you're participating in one of our natural history studies, then you'll see the tools that are used to select individuals for clinical trials, because those information are shared with drug developers to allow them to appropriately design clinical trials. But the main point, the main goal of those trials is to show that change in disease progression over as short a period of time as possible. So there's usually, we know that limb-girdle doesn't progress at the same rate. And so particularly, for example, on walking speed, if you get to a certain walking speed, you're more likely to progress over the next 12 months. And that's a key piece of information that's used to help select people for clinical trials. And then the other thing is just to say that because they're concerned about safety of that drug, they want to make sure that people with other medical conditions that might put them at increased risk aren't in, especially in those early clinical trials, which are really focused on safety.
A few more questions related to specific genes. Is there any gene therapy, or is there any therapy in development for limb-girdle in the MYOT gene?
Yeah, so the ones that have an active drug development program are the ones that I listed there. The tools that are used in those particular genes are applicable to almost any kind of recessive limb-girdle muscular dystrophy. There's some caveats there if the genes are too big. So Titin, for example, unfortunately is a very big gene and requires some additional technology. One of the challenges, I know the MDA is working with us on this, as is the NIH, is that for some really rare forms of limb-girdle muscular dystrophy, how do we get those going? How do we get those trials started without waiting for companies? And so you're gonna have to ask me that question like another year. We're working very hard on all the ultra rare forms.
Here's another question about a rare form. This attendee has limb-girdle R23 laminin, and how applicable is the knowledge gained on these more common limb-girdle types for the rarer ones? And you might have answered a little bit.
Yeah, so the reason why, I don't know if you could see, but in our consortium, our goal is to test the same outcome measures on every subtype for ultra rare forms like laminin. Laminin's a great example. Or I saw somebody at JAG2 or TCAP for these really, really rare forms of limb-girdle muscular dystrophy. We believe that we can take the information we've gained from the common forms of limb-girdle muscular dystrophy and translate that directly into those clinical trials. That's the goal. So, so I think across the board, we're trying to get it done for everybody on this call.
Is there a prognosis for patients that have neither confirmed gene mutations nor VOUS indications? So that 20 to 25%.
That's a hard question to answer because the prognosis is really associated with the gene mutation. The best way of thinking about it is, in general, even though I told you that it does change a little bit over time. In general, what has happened to you over the past 5 years is likely to portend what's going to happen over the next 5. So that's a good rule of thumb when you're thinking about prognosis. And then, please contact our consortium. We'll see if we can't help with that variant of unknown significance.
We also have another question about older limb-girdle patients. Is there anything that we can do? And maybe you want to talk a little bit about, what are the average age, gender of your clinical trial patients?
So gender is never an issue. We aim to recruit both men and women equally. For age, that depends a lot on the subtype more than but so particularly for like ANO5, that cohort goes up to 65 years old. And so there's definitely adults in our studies. It's probably more important to separate out whether or not you can walk 30 feet or not walk 30 feet. Mm-hmm. And we do, while most of the effort is focused on people who are able to walk, we do have sub-studies for people who aren't able to walk, because we think that as clinical trials move from phase 2 to phase 3, more people will be needed for those studies. And we want to see, does it have an effect on people who can't walk? And so we are thinking about that.
A question came in about how can we get a specific diagnosis for limb-girdle type?
for limb-girdle type. So you can
Yes.
go to your local MDA care center. There at that MDA care center, they should have the ability to order a free genetic testing panel. There's 2 different ones that are available to them. If they have any questions about that, they can reach out to me. They can reach out to Michelle as well, because that information is readily accessible to your local neurologist or physical medicine, whoever's running your care center. And that's the best first step. And so you'll either fall into one of those categories, and then if they need help interpreting that test again, they're always welcome to reach out to somebody like me. So.
Yes, also stay tuned because we do have a presentation on genetic testing and limb-girdle, and that's coming up at 1:05 p.m. Eastern time. So Caitlin Payne, a genetic counselor, will be able to answer all of your genetic testing questions.
Yeah.
Is heart failure being used as an endpoint?
That's a good question. So most of the effort is focused on the skeletal muscle because those endpoints are more accessible during the course of a clinical trial. We do measure heart failure, but it doesn't change as fast, which is good for your clinical care. It doesn't change as fast as your walking speed. And so it's captured, but will be unlikely to be kind of the top thing.
Thank you. One more question has come in. Any trials on FKRP?
Yes. So I mentioned that so our natural history study is close to enrollment for FKRP. There's a phase 2 clinical trial that's ongoing at BCU for that ribitol therapy. And I anticipate there are 2 publicly announced gene replacement therapy trials in FKRP. One is a company called AskBio, and the other is a company called Atomio, very similar sounding. Those trials have not begun, so I don't have details about who's going to be included or not included, but both companies have indicated that they plan to start those clinical trials within the year 2022. So we're about halfway through, so it's got to be coming soon. And if you have questions about that, again, you can email me. We will put you on our list to get you to a site for those trials as they open up.
Yes, Dr. Johnson mentioned clinicaltrials.gov. It can be a little tricky to navigate sometimes, so we do have a clinical trial finder tool on mda.org, and I'll just ask our tech person to put it in the chat. It's kind of an easier way to look for a clinical trial that you can be eligible. So I will ask our tech person to write it in the chat, and you can copy and paste it onto your browser. A question has come in on one gene mutation on the ADSSL1 in 1985. Is that still considered limb-girdle? Would it be important to maybe get another genetic test if there wasn't any, if you haven't had one since 1985?
Yeah, probably yes. The short answer is that the genetic testing, and you'll hear more about this afternoon, but the genetic testing has changed so much over that amount of time that it really has, it needs to be updated every 5 years if you don't already have a known diagnosis. So please go to your care center and talk to them about getting repeat testing.
Yes, absolutely. And if you're looking for a care center near you, Dr. Johnson said that I have all the answers, but I do not have all the answers on them, but our MDA Resource Center does. So if you want to give them a call or send them an email, they can connect you to the closest MDA care center near you, and I'll have our tech person also add the Resource Center to the chat, both the phone number. They're closed today, but they'll be back open on Monday. Or you can just email the Resource Center and they will get back to you. I think that may have been all the questions that we had. I don't see any more. All right, no more have come in. Well, thank you so much, Dr. Johnson, for taking the time on your Saturday to talk to us a little bit about research and clinical trials. We appreciate everything you do for the neuromuscular disease community. And thank you so much for being here today.
You bet.